Hello and welcome to this Active Motif webinar series. My name is Mason Brooks and I have with me today my colleague, Dr. Rwik Sen, who's a field application scientist with Active Motif, and he'll be talking about advances in chromatin and DNA shearing with the new PIXUL Multi-sample Sonicator and how it can be integrated into various types of research projects.
Thank you, Mason, for the introduction, and thank you to the audience for joining today. I'll be talking about PIXUL, a high-throughput sonicator, which sonicates up to 96 samples in less than 30 minutes with highly consistent results. It is economical, user-friendly, low maintenance, and portable, as you can see in the dimensions. And although PIXUL has been used a lot in chromatin and DNA shearing, but we also have consistent results in PIXUL applications for transcriptomics and proteomics experiments. This paper shows you the details of how PIXUL can be applied in a lot of different applications on the epigenome, genome, transcriptome, and proteome. But today, I will walk you briefly through how PIXUL can help you overcome the limitations of existing sonication workflows and how you can use PIXUL to address your research questions on the epigenome, genome, transcriptome, and proteome. The limitations in current sonication workflows are that they require extensive dedicated labor in sample preparation, in sonication, and downstream analysis. Some of the disadvantages are run-to-run consistency, and then you have samples from multiple separate tubes that need to be converted to a format that is compatible for downstream processing. So together, all these disadvantages and labor-intensive processes create a huge bottleneck. PIXUL can help you escape this bottleneck in conventional sonication. Why? Because PIXUL can use a plate where you can grow cells directly. This plate is a 96-well plate, which is TC treated, so you can either grow your cells directly on this plate, or you can transfer your samples on this plate and then put this in the machine for sonication, which takes less than 30 minutes. And then you can use the same plate to transfer to your analyzer machine for fragment analysis. So you can see that PIXUL is fast, PIXUL is easy to use, it is flexible because you can program up to 12 different simultaneous sonication conditions, and it is versatile because you can process a multiple type of tissue or cell samples. And then it is simple because it has an intuitive touch screen that I will show you later, and we have seen extremely high reproducibility or consistency in sonication that I will show you in the data slides later on, where we have seen that from 1 through 96 samples, there has been a very high consistency of the sonicated fragment sizes. And finally, it is really affordable because the consumables are not at all expensive. And I would remind you, as I showed you previously, that it is very portable and it can fit right on your benchtop and you can move it around across different benchtops in your labs. So before I show you the data, I would like to walk you through the PIXUL shearing mechanism. How does PIXUL shear your samples? You can see here that this is the 96 well plates, and beneath this well, there is an array of 96 concave lenses which focus ultrasonics that are produced by an array of transducers that lie further underneath. So if you take a closer look here, this is the transducer that produces acoustic waves which travel upward, and then the lenses help to focus these waves into the center of individual wells. And this is the reason you need to have a minimum sample volume in each well to be sonicated efficiently. Now, the resulting pressure of this standing wave generates a cavitation event which creates microscopic pockets of vacuum that results in the shearing of DNA. So this is how your DNA is sheared. I will quickly show you how the wells are sonicated. So what you are seeing here is the transducer assembly before it is installed into the machine, and then here the columns are being driven or sonicated synchronously in this pattern to minimize the heating while maintaining speed. And this is your touch screen. So here you can see it is a very intuitive touchscreen where you can assign up to 12 different sonication parameters and program them according to your 12 different columns. So what are some of the application areas that I mentioned? We have used PIXUL in genomic, epigenomic, transcriptomic, as well as proteomic analysis. So now let's move on to some of the PIXUL shearing data and the results. So first, we will start with shearing of genomic DNA. Here you can see the genomic DNA sonication products that we have got by using different sonication times and also by using different volumes or mass of sonication or input DNA. So as you can see that if we change the sonication time, then we see there is a shift in the fragment size of the sonicated DNA, but this shift is not very dramatic. Also, you can see that if you change the amount of your input DNA, you still don't see a very significant difference or a very drastic difference between the sonicated or sheared fragments. So this is very good that PIXUL does not mess up your sonication efficiency or the sonication size if the DNA concentration is varying. So you can work with a huge range of input DNA and still get consistent sonicated fragments. And you can also see that there is an control which clearly demarcates between your sonicated and unsonicated samples. Now next, I will show you here that PIXUL is highly consistent. Here we have the sonicated fragments, the results from 1 through 96 wells that were sonicated, and each of the fragments are highly consistent. Their sizes range between 350 to 400 base pairs. And you can see that the unsonicated controls are starkly different from these ones. So you can see that there is a very high level of consistency between 1 through 96 wells in the PIXUL sonication. Now we will move on to see how PIXUL helps in shearing RNA. Here you can see the sonicated products from the triazole-treated RNA in 3 different kinds of buffer conditions. And you see that there is a high range of similarity or consistency in all 3 conditions, which is starkly different from the unsonicated condition. Next, we move on to chromatin, because chromatin-related research and services are a priority of Active Motif. So we have done a lot of chromatin-based assays using PIXUL. So here you can see that the size of the sonicated chromatin samples are again very consistent, irrespective of the amount of starting material that was used. So no matter what we use, if it's a 10,000 cell sample or if it's a 1 million cell sample, we always get a very high consistency of the size of the sheared fragment. So this is very important if you are trying to look at histone post-translational modifications or transcription factors in your chip assay. For histone post-translational modifications, you need a low number of cells, whereas for transcription factors, you need a high number of cells. And you can see here that for both conditions, you will always get a very similar size of sonicated fragments. So you can maintain the same protocol irrespective of your cell count. And this is gel electrophoresis that shows again how highly consistent the PIXUL sonicated fragments are. So now let's take a deep dive into the Chipseek data that has been generated by PIXUL. Now recently, there has been a lot of interest in combinatorial histone post-translational modifications, because studying these combinatorial post-translational modifications gives you a whole lot more information than studying individual post-translational modifications. And this is one of the nice things about PIXUL, because it allows you to compare large-scale comparison between sample types and modifications as to what is going on in your cell or tissue sample. It gives you a complete picture. Here we are looking at Chipseek data for histone H3K27 acetylation across different tissue types obtained from mice. So here, not only can you compare how histone H3K27 is distributed in these three tissue types, but you can also see that the amount of starting material is not affecting the output that you are getting from PIXUL. So ranging from 1 milligram of tissue to 30 milligrams, you are always having consistent pattern of histone H3K27 acetylation. And this also lets you compare your marks across different tissue types. Here, we are looking at various kinds of histone modifications across different cell types. So you're looking at histone H3K4 methylation, K9 acetylation methylation, as well as K27 acetylation methylation, and K36. And the three cell lines that we have used here, GM12878 is treated as a control, and GM08714 is ICF. This is a disease known as immunodeficiency centromeric region instability facial anomaly syndrome. In this disease, most of the patients have mutation in DNAMT3B, and this disease is characterized by rearrangement-prone heterochromatic regions which exhibit DNA hypomethylation. And for the Pfeiffer cell line, here we have a mutation in EZH2, which is a histone H3K27 methyltransferase. So you can see here that the H3K27 trimethylation peaks are absent in this cell line compared to the other two. So this kind of analysis lets you appreciate all these different histone marks across the total genome, and so you can know what's going on for a broader scale of epigenetic changes. And since this is a lot of data to capture here, we use a software called ChromeHMM. So ChromeHMM is a software that allows for the construction of hidden Markov models, and these help you to model the presence or absence of each chromatin mark to annotate a genome. And what it tells you is that the underlying epigenetic state of each particular region based on the combinations of all the modifications that it obtains from the multiple chromatin datasets like CHIP and ATAC-seq. And what it gives you is that these resulting tracks, which are shown here, and each color here is a classification that is associated with a particular epigenetic state. So in this kind of analysis, things are a lot easier to compare about what is happening across the genome across different cell types. And then finally, I'll talk about the multi-omics experiments that we can do with PIXUL. The first experiment that we see here is CHIP for RNA polymerase II and RT-PCR for mRNA expression. So as you can see here that in males and females, they have analyzed the association of RNA polymerase II and also looked at the corresponding mRNA expression, and both of these are pretty consistent and complementing each other as expected. Where you have fault association, there you are seeing an expression of the mRNA. So again, you can get consistent analysis for CHIP and mRNA, or rather your epigenome and transcriptome. This experiment shows you how you can use PIXUL for multi-omics analysis for epigenomics, transcriptomics, and proteomics. So this is about the gene EGR1, which is an early growth response gene that is induced by serum. So this region here is a CTCF binding site that is a zinc finger chromatin binding transcription factor binding site, and RNA polymerase II CHIP was analyzed using primers designed along this region. So you can see that after treating with serum for this much time, there is an association of RNA polymerase II that corresponds with the transcriptomic or mRNA expression of that gene, and which further corresponds to the downstream protein expression of EGR1 after 30 minutes of serum treatment. This slide shows you how you can use PIXUL for multi-omics analysis, and currently our collaborators that are using PIXUL are working on all these areas, and even though I couldn't show you more data on proteomics, but there is work in progress that can soon be published or made available to you regarding proteomics. So this brings us to the conclusion that PIXUL is consistent, simple to use, fast, flexible, affordable, and versatile. And I hope I have been able to convince you about how useful PIXUL is in addressing the questions of your research in the field of epigenomics, genomics, transcriptomics, and proteomics. Thank you very much for your time and attention. For more information on PIXUL, please visit us at this link or contact us here for more information. And please stay tuned for more exciting research news from Active Motif. Thank you.